The leaf. Introduction. The flat, green and broad part of the shoot located on the node of the stem is called leaf. The green color of leaves is due to the presence of a green pigment called chlorophyll, which has the ability to convert light energy into chemical energy, glucose, and thus helps during photosynthesis. Leaves do not keep on growing continuously like the stem. Instead, their growth stops on attaining full size. Leaf is lateral, flat, thin, green, and broad outgrowth arising at the node of the stem. The lowermost leaves are darker, broader, and older, whereas the upper leaves are small, young, and bright green. It is light-gathering part of the plant. It contains green-colored pigment, chlorophyll, for photosynthesis. There is always a bud on the axil of a leaf called axillary bud. Axil is the angle between the leaf and the stem. Like stem, they do not grow continuously but stop growing on attaining full size. Parts of leaves. A typical leaf has following parts: lamina or leaf blade. It is the green, flat and broad part of the leaf. It is well adapted to absorb sunlight and carbon dioxide for photosynthesis. Leaf base. It is the part of the leaf in contact with the stem at the node. Petiole or leaf stalk. It is the stalk by which the leaf lamina is attached to the main stem. Midrib. The petiole enters into the lamina of the leaf and continues as the midrib. Veins. Midrib gives out numerous fine branches called veins. Veinlets. The veins divide and subdivide to form a fine network of veinlets which keep the leaf in expanded form. Leaf apex and leaf margin. The tip of the lamina is called leaf apex and the edge on all sides is called the leaf margin. Types of leaves. Types of leaves: simple leaves, compound leaves. Simple leaves. The leaves in which the lamina is undivided and is a single piece are called simple leaves. In these leaves, various margin incisions, if present, never touch the midrib. Examples: mango, jamun, peepal, castor, cotton, China rose. Compound leaves. The leaves in which lamina is divided into small segments which are attached to midrib, rachis, and its branches or tip of petiole are called compound leaves. In these leaves, incisions reach up to the midrib or petiole. Due to this, each individual leaf looks like a group of small segments called leaflets. Examples: rose, gram, neem, pea, tamarind. Venation. The arrangement of veins and veinlets in a leaf is known as venation. It is of two types. Reticulate venation. When veins and veinlets are irregularly distributed in the lamina of the leaf, forming a network, it is called reticulate venation. This is commonly found in the leaves of dicot plants. For example, peepal, mango, guava. Parallel venation. When veins and veinlets run parallel to each other towards the tip of the leaf, it is called parallel venation. This is commonly found in the leaves of monocot plants, for example, sugarcane, wheat, maize, banana, bamboo. Phyllotaxy. The arrangement of leaves on the stem is called phyllotaxy. It prevents overcrowding among leaves and helps in better exposure of leaf to sunlight and air. It is mainly of three types: opposite, alternate, and walled. Opposite phyllotaxy. When two leaves arise on each node opposite to each other, the phyllotaxy is said to be opposite. For example, tulsi, jasmine, guava. Alternate phyllotaxy. When single leaf arises from each node and the next leaf arises from the successive node in opposite side of previous leaf, it is called alternate phyllotaxy. For example, rose, castor, peepal, china rose, sunflower, mint, mango. Walled phyllotaxy. When more than two leaves arise at each node arranged in a wall or circle, it is called walled phyllotaxy. For example, oleander, asparagus. Functions of leaf. Photosynthesis. The process by which green plants prepare their food with the help of carbon dioxide and water in the presence of sunlight and chlorophyll is known as photosynthesis. Exchange of gases. The leaves on their surface carry tiny pores called stomata, which help in exchange of gases for photosynthesis and respiration. Transpiration. The removal of excess of water from the plants in the form of water vapors through stomata is called transpiration. Modification of leaves. Along with the important vital function of leaf, that is photosynthesis, leaves of certain plants get modified to perform some additional functions to cope up with the needs of environment. It is of following types. Leaf tendril. In some weak-stemmed plants, either the whole leaf or leaflet may be modified into wiry coiled structures called tendrils. As they touch any object, they coil around it and support the plant to climb up. For example, sweet pea, gloriosa, smilax. Leaf spines. The leaves are modified to a hard pointed structure called spines. These These spines check the loss of water from plant body, helping the plant to conserve water. They also protect the plant from grazing animals, for example, cactus, Obchundia, berberis, aloe. Phyllodes. When the petiole becomes expanded into a green, flat, leaf-like photosynthetic structure, it is called phyllodes. It helps in conservation of water and reduces water loss. For example, Australian acacia. Hooks. In bignonia, terminal leaflets become modified into hooks for climbing on the support. The hooks give the appearance of the claws of a cat. Scale leaves. These are dry and thin or thick and fleshy, non-photosynthetic leaves which take on a protective role. They protect the buds. For example, ginger, onion. Fact file. In Opuntia, prickly pear, the leaves are modified into small, hard, conical spines. In aloe, prickly poppy, the leaves have spines on their margins. In berberis, all the leaves are modified into spines. In the date palm and dagger plant, only the apex of a leaf is spiny. The leaf tip forms the lid and the petiole becomes a long, thin, coiled structure supporting the pitcher. Along the edges of the opening of the pitcher are placed small hooks. An insect is trapped in the pitcher and digested by the secretion of enzymes into the sac. The slender structure at its lower end is a modified leaf base. The leaf base is green, flattened and photosynthetic. These are found in Garo and Khasi hills of Meghalaya. Insectivorous plants. These plants grow in infertile soil, where they are deprived of nitrogen, which is essential for protein formation and growth. Therefore, these plants trap and digest insects to get proteins from their development. These are as follows. Pitcher plant. The leaf lamina becomes modified into a pitcher to capture insects. Bladderwort. In this, some of the leaves are highly segmented and modified into a sac-like structure called bladder. The bladder has an entry point which can be closed. The insects enter through this opening and are digested by the secretion of enzymes. Venus flytrap. In this, the leaf blade is divided into two parts, hinged along the midrib. The two parts get interlocked by marginal teeth and prevent the insect from escaping. The insect is then digested by digestive juices secreted by the plant. Vegetative propagation in leaf. In some plants, leaves are modified to bear foliar buds, from which new plants grow and develop. For example, bryophyllum and begonia. The leaf in the bryophyllum plant is broad, has notches at its margins. buds arise from the notches. These buds are called epiphyllous buds. When the buds drop off from the leaf and are placed in soil, they develop into new plant with roots and small leaves. The vegetative propagation in begonia is also in a similar way.